Good afternoon, everybody, and thank you so much for joining our educational webinar today. And apologies for just starting the meeting a few minutes late. I know that some of you have been waiting for over 30 minutes already, so I love to see how eager everybody is to learn more. Let's start off today's presentation. I just need to find my own slides here. There we go. This is, of course, about HLA-DPB1 permissiveness. The session or this webinar will be recorded. Afterwards, it will be available to all WMDA members on WMDA Share. If there's any technical issues or support, both myself and my colleague Hans-Peter are here to help you. Please drop that in the chat function. There will also be a Q&A towards the end, time permitting. So if any questions you come up during the course of this webinar, please put them in the Q&A and we will address them at the end. And then we would like to thank our generous sponsor, NextGen, for being our webinar sponsor, at least until the middle of 2026. They were also supporting us throughout 2025. So thanks for that. The agenda briefly is we'll start with some icebreaker polls. Then Esteban will take us through a brief history of HLA-DPB1. Stephen is going to give us a bit of insight on the clinical relevance of HLA-DPB1 in donor selection, and Dominic will then take us through some of those permissiveness tools that they've built at the IPD-IMGT. And then, of course, as I said, time permitting, we'll have some Q&A towards the end. Stephen, I'd like to give you an opportunity to introduce yourself to us, please.
Thanks, Alicia. Good afternoon. Good morning, everyone. My name is Steven Spellman. I go by Steve to all my friends, which I consider you each. I am the Vice President of Research at the CIBMTR and Clinical Services here in Minneapolis, Minnesota, and I am a Senior Scientific Director within the CIBMTR, and glad to be here today.
Thank you so much. And over to you, Dominic.
Yeah, thank you. I'm Dominic Barker. You can call me Dom. I'm a bioinformatics scientist working on the IPD-IMGT HLA database at the Anthony Nolan Research Institute. And I was the developer of the recently released new DPB TCE tool on the database website in the last year.
Thanks, Dom. And last but not least, Esteban.
Thank you very much, Alicia, for the invitation. My name is Esteban Arrieta-Bolanos, and I work as a researcher at the Institute for Experimental Cellular Therapy in Essen, Germany. And yeah, I'm delighted to be here. I'm happy to be here with Steve and Dom, who are not only colleagues, but also friends.
Brilliant. Thank you so much for also agreeing, all three of you, to present to us and our members today. As I mentioned, we would like to also then take just a quick poll to get a good understanding of where you, as our participants, are in terms of HLA-DPB1. So if you can grab your phone, you should be able to scan this QR code, and then I will share the outcome of the next few polls with you. You will have like a minute or so just to answer the questions that come up on the screen, and they're completely anonymous, so you don't have to, you shouldn't be asked to fill in your name. This is really just to get a broad idea of where we are in terms of the membership and what it is you'd like to learn more. So I'll share that on the screen right now. The first question is, do you consider HLA-DPB1 in donor selection? See, we have quite active participants. Thank you so much. I'll wait another few seconds for more votes to come through. I see that already over 90 people have voted in this first poll. There is a clear winner here with 60% of the participants saying that they always use or consider HLA-DPB1 in donor selection, which is brilliant then to have you here. So hopefully today you can learn a little bit more about the impact thereof and how to further improve your knowledge and intuition when it comes to HLA-DPB1. Right, shall we go to the next question? It should pop up on your screen now, but the question is, if or when you consider HLA-DPB1 in donor selection, do you use version 2 or version 3? Voting is now live. For a little bit of context, version 2 is the version that is currently used in the WMDA search and match service. Version 3, we are aware, have already been implemented with some laboratories in the world. And so you might be receiving version 3 results from your local laboratory. If you're unsure, of course, you can also just say, I don't know. All those currently voting for version 2, this is brilliant because you can learn all about version 3 from Esteban in just a minute. Then let's go to the last question. How familiar are you with the impact of HLA-DPB1 permissiveness in hematopoietic stem cell transplantation? And of course, if you feel like you are not familiar enough, you are in the right place. Hence the last option, that you're unfamiliar, and that's exactly why you're here. In the interest of time, I'm going to stop the voting right now. Thank you so much. I think that that has really helped myself, WMDA, and also our presenters just give a bit of an idea of where we are in terms of the audience that we're addressing today. With that, I would like to hand over to you, Esteban, and I'll stop sharing my screen and you can share your screen and start using your slides.
Esteban, it seems you're muted.
I'm sorry. I am sharing my slides now. Can you see them correctly?
Yes,
Yeah,
thank you.
Excellent. Thank you very much. Right, so let's get started. So today I was asked to prepare a little overview of the history of the T-cell epitope model for HLA-DPB1 permissiveness in hematopoietic stem cell transplantation, with a focus on the newest developments and what's behind the new tools that Alicia already mentioned. So here's a short outline of the talk. Essentially, I would like to remind you why we care about HLA-DP, what the T-cell epitope model is, and what the evolution of it has been during the last 20 years or so, and then delve into the latest data that supports some of the changes that we have implemented, both clinically and from a biological point of view. And finally, to mention the implementation of these changes to this tool that our colleagues at Anthony Nolan maintain. I will not go into the details as to how to use the tool because Dom will cover that after Steve actually gives the bigger picture of HLA-DP mismatching in transplantation nowadays. So just to start, one thing I wanted to remind you is that HLA-DPB1 is an HLA molecule, obviously, that has been known by us for a long time, but it has certain things that make it a little bit different from the rest of the loci that we investigate in this field. And you might remember that usually when you look at haplotypes of HLA, There's a tight linkage disequilibrium. There's what some people call conserved haplotypes. And so we know that these variants are passed together. However, for HLA-DP, this is a little different already from the start. Its location in chromosome 6 is much more centromeric than the other loci. And the main thing is that between HLA-DP and the other HLA loci, there are at least 3 recombination hotspots, which you can see here in this cartoon. And that reduces the linkage disequilibrium between A, B, C, DR, and DQ and DPB1. So that gives another layer of variability in terms of what you can find in human populations and patient and donor populations. HLA-DP is also a little bit less polymorphic than other HLA loci, although this is already being challenged a little bit because of the increasing description of new alleles with the new technologies, but still is relatively less polymorphic. And we have in human populations usually a handful of alleles that really dominate the genetic profile of those human groups. So that's also an interesting feature about the HLA-DP. HLA-DP is nevertheless a classical transplantation antigen. It was discovered and shown to play a role in transplantation. And it does so by doing what HLA molecules do as their job. It presents antigens in forms of peptides to cells of the immune system, which, depending on the matching between you as a patient or as a donor and the other individual, can elicit a strong immune response that can lead to complications after transplant, but also that it's involved in the beneficial aspects of transplantation as a therapeutic approach for hematological cancers. As you know very well, we consider loci A, B, C, and DR in North America and DQ also in Europe as the main loci to define the compatibility between patients and donors in this context. And HLA-DP was like a latecomer to the party. It was discovered a little bit later, and people, for different reasons, they didn't include it in the selection. I think one of the main reasons is that, of course, the lower linkage disequilibrium posed an important barrier to finding 12 out of 12 donors, at least at that time. And so it was not officially incorporated into this high-level compatibility group. However, early on, already more than 20 years ago, it was recognized that it also had an impact. So mismatches at HLA-DP also had an impact on transplant outcome. And what was really interesting from these early studies, it was that HLA-DP mismatches had an interesting property of associating with reduced relapse in malignant disease transplantation. And that came at a cost of increased GVHD. So it was never really possible to use it intelligently until we understood a little bit more about its biology. And this is going to be something that has been important at different stages of the TCE model, and it's actually important in the latest developments of it. So another interesting thing is that because it has been ignored a long time as a transplant antigen that has to be matched, there's a lot of mismatching in clinical transplantation. As you know, most of the transplants are performed across HLA-DP variants, the unrelated transplants. And this has changed a little bit, I have to say, because probably of people being more aware of HLA-DP and better technologies, better algorithms. The thing is that the matching for HLA-DP has increased from essentially 15% previous to 2011 to almost 31% now in the EBMT registry. And that goes to show that people have increasingly taken HLA-DP into account and have looked actively for matching at this locus when possible. That among the 10 of 10 reaches almost 40%, which I think is a big change compared to just a few years before. And what's also interesting about the mismatches at HLA-DP is that they are really highly skewed and concentrated towards a few combinations of highly frequent alleles. So that makes matching at this locus also different from matching at other loci. Again, this has to do with the allelic frequencies at HLA-DP in the different populations that we work with, but this is a key feature also of HLA-DP mismatches in clinical transplantation. And so when people started to try to understand how the mismatches at this locus could be interpreted and could be used clinically, it all started with what we now call the T-cell epitope model. In that model, essentially, the researchers in Milan were able to isolate clones from a patient who was suffering a rejection of a transplant that was actually mismatched for HLA-DP. And so, interestingly, when they took those cells and isolated them and expanded them in vitro, they were able to analyze what patterns of recognition they had against the HLA-DP molecules. And just compressing at least 10 to 15 years of research in one slide, this was the basis of our understanding of the T-cell epitope model, because some of the antigens, some of the alleles, were recognized by all clones isolated from that patient, and also T-cell lines generated afterwards, whereas other group of alleles were only partially recognized. And then there was a bunch of other alleles that were not recognized at all. And that was the basis for the classification of the DPB1 alleles into 3 groups that were functionally different. This led to a classification of mismatches across groups as non-permissive and mismatches within the groups as permissive, which was shown in different studies at the national, international, and global level to have an impact on transplant outcome in that era. This is just one of the largest studies, but it was confirmed in other studies also from CIBMTR, and this established the T-cell epitope model as a useful tool for assessment of mismatches at this locus in transplantation. Again, one interesting feature about HLA-DP and about the the TCE model itself, it was that it gave us the possibility to isolate certain mismatches that had a specific or special relation with respect to relapse and non-relapse mortality. So in this case, permissive mismatches were repeatedly shown to reduce relapse risk without increasing non-relapse mortality to the levels that non-permissive mismatches would do. That's what you can see in this study in these green curves here. So the permissive mismatches played like this dual role, which in the end was deemed to be probably beneficial for many patients in terms of their outcome. But the group in Milan and later here in Essen were really keen on not just describing the clinical associations, but going deeper into understanding the biological principles of this phenomenon. And this piece of work here by my colleague Pietro Clirello was fundamental in this aim, because he went through the painstaking process of mutating one of the HLA-DP molecules at different positions with different amino acids and analyzing what the impact of those changes was for the allorecognition by T cells. And this is all very interesting from a immunobiological point of view, but it also had a practical usefulness in that this was actually the basis for what we later called the functional distance between HLA-DP alleles. So based on the impact of these molecular changes on the HLA-DP molecule, he was able to derive the score that allowed us to classify the alleles, and he showed that this functional distance was correlated with the previously defined TCE groups. But what was most important from this work was that this allowed us to classify alleles old and new without going to the lab and performing cumbersome cellular experiments that were probably not feasible. They would certainly not be feasible today when we have thousands of alleles. And so this really allowed us to classify every single HLA-DP allele into the respective group and then provide information on transplants that were in which these alleles were involved as mismatches. I'm showing you this slide again because that was not the end of that story. Actually, this opened a new avenue of research. As soon as we realized that most of the residues that had strong implications with respect to the impact on alloreactive responses, which you can see here on the right side of the slide, were actually at residues that seemed to, or were predicted to have a role in the binding of the peptide of the HLA-DP molecule. And that led us to think about the possibility that the peptides being presented and their diversity could be something that actually could explain the deeper basis of the T-cell epitope model. And we pursued this hypothesis by trying to correlate what we knew before from the early experiments, that permissive mismatches in vitro led to limited alloreactive responses, whereas non-permissive mismatches led to very strong responses. And when we looked at the peptidome, so the repertoire of peptides presented by those HLA-DP allotypes, we noticed that the overlap, so the sharing of peptides within the group, the alleles, or between the alleles within the groups that we had described before, was significantly higher than the background, suggesting that this immunopeptidome overlap was at the basis of permissiveness for HLA-DP mismatches. We later on, when we went on back to the lab, and then showed that this actually had an implication for alloreactive responses in vitro. I'm not going to go into all these experimental details. This can be found in the respective publications, but it really showed that this was at the basis of what we had observed before, and that we could also even manipulate this by changing the immunopeptidome, increasing the T-cell receptor diversity and increasing the immune responses as a whole against HLA-DP allotypes that would have been otherwise permissive and low responsiveness. We did also further experiments where we showed that the degree of immunopeptidome diversity is fundamental for alloreactivity in general, but especially in this case for HLA-DP and the model itself. And we showed that the editing of the immunopeptidome by the non-classical molecule HLA-DM was key to explain the observations that we had made in vitro regarding the strength of the alloreactive response. Again, all this is immunobiological, so I just wanted to point to the relevant publications. If you're interested, you can, of course, have a deeper look. But what's really important about this is that this, again, was something that we translated to the clinical practice. And actually, it resurfaced one concept that had gone a little bit under the radar. It was actually published by Katharina Fleischauer a few years before we even made the discoveries with the immunopeptidomes. And it's the concept of immunogenicity versus diversity of the TCE model. And why I'm going to make an important point about this is because this is one of the reasons why some of the versions of the tool changed, and some of the transplants that you might, or the typings that you put in, might give you different results depending on what tool you're using. So basically, the original concept of the TCE model was that there was a group of alleles that was more immunogenic. This was based on the cross-reactivity experiments from the early days. However, another possibility from a biological perspective was that this was not just because some were more immunogenic than others, but it was just because they were diverse, they were different. I mean, we know that the alleles are structurally different, and then we confirmed that functionally, through the peptidomes, this was the case. And so the concept is not that we have a group of alleles that is supposed to be at the high end of immunogenicity, but that the immunogenicity of the mismatch depends on the diversity of the functional characteristics of the HLA-DP models. To put it in practical terms, you can see some examples here. Essentially, you can have transplants that have a bidirectional immunogenicity, meaning that there's going to be a mismatch in the HBG and the GVH direction that is of potential clinical relevance, whereas the other model essentially didn't consider this possibility. And so the old model was based on the immunogenicity, and that meant that there was no bidirectionality. Now, the more established model, which is supported by the peptidome data, is the one that we call the diversity model. And as Dominic will show you later, that is the basis for version 2.1 of the tool. And we can look at some examples if you want later on. Yet another important development recently is that all this was great from an experimental point of view, a scientific point of view, but there was an important limitation in that everything that was calculated before in terms of functional distance and all these comparisons was always done taking DP9, so this allotype as the reference. So that led to essentially a unidimensional calculation of these distances between the alleles. But if you look at it from a structural point of view, you can see that DP9 and other alleles might be at similar distances, but between the alleles of group 3, for example, here you can see DP1, DP5, and then the DP2 and DP4 groups, they can also have a big distance between themselves, which is not captured necessarily by the functional distance that is calculated from just the DP9 allele. And actually, if you look at this, from an immunopeptidomic perspective, this is supported also by the peptide repertoires. What you see here are peptide binding motifs from different HLA-DP allotypes, from different TCE groups. And as you can see for TCE1 and TCE2, the representative alleles are very similar in terms of their peptide binding properties. That means that they are functionally related, and probably that's why they are permissive amongst themselves. However, for TCE group 3, we found that there was a lot of diversity here also in terms of the peptide binding characteristics of these molecules. You can see that here comparing O201 and 1301, for example, there's essentially no sharing in the PBM, the peptide binding motif between those two group 3 molecules. We went, as always, back to the lab and we tested this experimentally, and we confirmed that when we exposed responders in what we then called a non-core scenario of mismatching between TCE3 alleles, those alloresponses were intermediate between the non-permissive mismatch to the right here and the traditional permissives, or what we then call the core permissives, to the left. So this gave also a functional immunological support for what we expected to be a functional diversity within group 3 alleles. And now the question came, does this make any difference in terms of clinical outcome, right? So we went back to the clinical studies, and then we, in collaboration with the CABMTR, who had this really nice cohort of over 5,000 transplants performed mostly in North America, we were able to show that the risk of acute GVHD actually was stratified very nicely according to whether we had a match for DP, a core permissive mismatch, or a non-core permissive mismatch. And actually, from a statistical point of view, the only group of permissive mismatches that had a lower risk of acute GVHD were the non-core, so the core permissives, sorry. Whereas the non-core permissives did not differ from the non-permissives. In a subsequent study, we tried to investigate this a bit further. First of all, we wanted to confirm that what we observed in the North American cohort was applicable to a cohort from European transplants. And also we postulated an additional hypothesis that maybe the position of the non-core allele could play an important role, giving the mismatch constellation a direction. And we did this in a collaboration with the EBMT in a larger cohort. Over 8,000 pairs were included in this study, and then we tested the association between our core and non-core mismatches and transplant outcome. And what we were surprised to find was that, first of all, we were able to confirm that core permissive mismatches are really the ones that minimize acute GVHD risks in HLA-DPB mismatch transplantation. They do not differ statistically from the matched pairs, whereas some subsets of the non-core permissive and definitely the non-permissives did have higher risks for acute GVHD, as you can see to the left of the slide. But what was interesting also in this study was that the non-core permissives in the GVH direction did have a significant reduction of the risk of relapse for patients with hematological malignancies. And what gave us also the hope that this could have a practical usefulness was that when we looked at the non-relapse mortality risk for both the core permissive and the non-core permissives, neither subset had a significant increase compared to the matched pairs. So this opened the possibility for what we are now putting forward as an idea to use directional permissive mismatches according to the main clinical goal that you have. On the one hand, if your goal is to really minimize acute GVHD, and you cannot use a matched DP matched donor, then you might as well try to match and end up with a core permissive mismatch. Whereas if you are worried that your patient has a high risk of relapse, you might be able in some cases to select for an HLA-DP mismatch of a little bit increased immunogenicity with increased alloreactive responses and lower relapse risk by selecting a non-core permissive mismatch. And this has been then the basis of the changes in the algorithm. Just to show you briefly how this looks in practice, in real world data, the frequencies of these mismatch subsets. From all 10 of 10 matched patients, which you can see to the left of the slide, in the EBMT cohort, about 37% will have a 40% will have a permissive mismatch. And from those, then the 37% will have a TCE group 3 permissive mismatch. So this goes to show how frequent these specific mismatches are. So not all mismatches that are permissive are equally frequent. TCE group 3 permissive mismatches are by far the most frequent. And when you look at the subsets of those permissive mismatches for group 3, you will see that it distributes nicely, almost 50-50 between the core permissive and the non-core permissives. When you look at the directionality of the permissive mismatches to the right, you see that from the non-core permissive mismatches, you have a fair distribution and essentially 26%, so 1/4 of the permissive mismatches in the EBMT cohort were GVH in their direction. So that goes to show that these are really frequent instances in clinical real-life transplantation. I'm getting to the end of my presentation. As I said, Dom is going to speak about the tool in depth, so I just want to show you how version 3 looks. And maybe the most important thing is to the right, because with Dom and James Robinson's help, we have been able to apply the classification of all TCE, of all DPB1 alleles, according to their, not only their TCE grouping, but also their subgrouping into core and non-core alleles. So that's been done. In this case, this is a distribution for the version of the database that was of over 1,600 proteins. But the most important thing is that they are now doing this prospectively. So every new allele that's discovered, it's immediately assigned a core, non-core label according to the set of rules that we worked out with them. Finally, you might be aware of course, the other models to assess HLA-DP mismatches in clinical transplantations. The transplantation here, I think what's important to highlight is if you want to look at the expression model, for example, there's a lot of correlation or overlap between the TCE and the expression model, as you might know. But what's nice about the core-non-core model is that it really, first of all, it can be applied to all pairs, whereas the expression model can only be applied to about 60% of the pairs because it's specific for single mismatches in the GVH direction. But also, if you look at the overlap between high expression permissive mismatches and the non-core GVH mismatches, this is very good. And it actually adds information because, like I said, in many instances, the expression model cannot be applied by definition. But this information can be collected also in the core non-core TCE model. So this is, I think, an advantage also of using this, as it gives you also indirectly the relation with the expression model. Finally, I would like to thank my colleagues at the Institute for Experimental Cell Therapy in Essen, our collaborators at CA-BMTR and EBMT, who facilitate all these clinical studies, also in Leiden and Bochum, where they helped us a lot with the immunopeptidomics analysis, and clearly the people at Anthony Nolan and WMDA for the invitation. I will be happy to answer any questions you have at the end of the presentation. Thank you.
Thank you very much, Esteban. Then we will move on to you, Steve, if you'd like to share your screen.
Sure. Thanks, Alicia. And thank you, Esteban, for that amazing tour de force biological overview of all of the amazing work that your team has done to really characterize the differences here. So, all right, just make sure I am sharing appropriately.
Yes, got it. Thanks.
All right, great. So I'm going to switch gears a little bit here and focus mainly on the clinical relevance of HLA-DPB1 TCE matching in donor selection. And I will be pointing out the various algorithms that have been used in some of these analyses just for context for the team. So by way of outline, I'm going to focus on the background on HLA-DPB1 and TCE matching in allogeneic transplant, really kind of telling us the story for how this evolved over time. And then what are we seeing today? We have, at least in the U.S. and I know in Europe and other countries, we're starting to see quite a large paradigm shift towards new GVHD prophylaxis strategies, mainly the utilization of post-transplant cyclophosphamide, predominantly in the mismatch setting, but also in the match setting nowadays, and the impacts that has. And then go through two quick cases just to kind of illustrate how HLA-DPB1 TCE matching should be incorporated into donor selection algorithms. So this is the seminal paper from the CIBMTR looking at high resolution matching in allogeneic transplantation that was published in Blood back in 2007 in a study led by Dr. Stephanie Lee from Fred Hutch. And what this demonstrated was that at 1 year and even out to 5 years, what we saw with each mismatch at HLA-A, B, C, or DRB1 was about a 10% decrement in overall survival with each of those mismatches. Now, this is using traditional calcineurin inhibitor-based GVHD prophylaxis. So whether that's cyclosporin or tacrolimus along with methotrexate typically. Now, in this population, we did evaluate HLA-DP only at the allele level. This was prior to much of the description of the TCE epitopes, and we did not see an association with any outcomes with the exception of some increased risk of acute GVHD grade 2 to 4. No associations with any survival outcomes. That also applies to DQ, and that's why I'll be talking about matching mainly in the context of 8 of 8. DQ mismatches do not associate with survival outcomes in CIBMTR studies, and so we tend to put that aside to not create any confusion about the importance of that mismatch. Now, looking at HLA-DPB1 TCE T-cell epitope matching, this was one of the first large studies that was completed through the International Histocompatibility Workshop that was led by Katharina Fleischauer and Bronwyn Shaw and Effie Petersdorf as the lead for the HCT component. And what that demonstrated was using version 1.0 was HLA-DPB1 matched and permissive mismatch led to better survival outcomes compared to non-permissive HLA-DPB1 mismatches. And this is in the context of 10 of 10 or 8 of 8 or 9 of 10 matched, mainly mismatches at HLA-A, B, C, and DRB1. We took this forward and validated these findings in a study from the CIBMTR that was led by Joseph Padala and published in Blood in 2014. This was a follow-on from the Lee study. The Lee study included about 3,800 transplant pairs. The Padala study included about 8,000. So much larger cohort, but still seeing some benefits of permissive mismatching. Now, we wanted to take this forward and understand how T-cell epitope matching mattered in the context of broader selectable factors that can be modified in donor selection. And so in a study that was led by Bronwyn Shaw through the CIBMTR Immunobiology Working Committee, we attempted to generate a donor selection score. So we used existing validated data sets. These are pretty historical at this point, but 1999 to 2011, all of them were 8 of 8 matched using high resolution methods, split into training and testing. And when we first started with the training set, we did see an effect of DPB1 TCE, again at the 1.0 model, donor age, CMV match, ABO match, but this failed under validation in the test cohort. We then pulled out another data set from the CIBMTR that was more contemporary at the time, so 2012 to 2014, and about 4,500 cases. None of those scores validated, and the only thing that was associated, the only donor factor that was associated with overall survival, which had been described many times in the past, was an increased risk associated with increasing donor age. And this just demonstrates the linear impact of donor age that was seen in this model. And so that was our takeaway there. These results were incorporated then into the NMDP CIBMTR donor selection guidelines that were published in 2019 in Blood, led by Jason Dean and Joseph Padala. And the table here illustrates the importance that we were putting on these factors. So high resolution matching at HLA-A, B, C, and DRB1 should be your first selection criteria, but then donor age. So selecting for younger donors and then potentially considering permissive mismatching at HLA-DPB1 as well as other factors. Now, we wanted to create a bit more precision around those cutoffs around donor age. This has been described as, well, maybe we make that cut point 30, maybe we make it 35. We wanted to determine with a little bit more accuracy and clarity what those cut points actually happen to be, as well as, again, looking at all of those other non-modifiable factors in the donor that could be optimized in the selection process. So here we consider 2 time to event outcomes separately with their trade-offs. So the first being overall survival, and the second is event-free survival, and that was defined as death, relapse, graft failure, rejection, or moderate to severe chronic GVHD. You may note that acute GVHD was not included in this definition, and the reason that we left it out is that acute GVHD is a transient state. If that doesn't result in relapse for overtreatment of immunosuppression or death or transition into moderate to severe chronic GVHD, the study team felt that that was less critical to consider, and so we focused on event-free survival. We also used a more advanced machine learning regression methodology based on work done at the Medical College of Wisconsin, led by Brent Logan and Rodney Sparapani, using non-parametric failure time Bayesian additive regression trees. I'm not going to go into that methodology too much here. So our study cohort for this was a very contemporary cohort of transplants from reported to the CIBMTR in the US, first allo transplants completed between 2016 and 2019, including all diseases. The vast majority of these, so about 90%, were calcineurin inhibitor-based GVHD prophylaxis. This was limited to matched unrelated donors, defined as high-resolution matched at HLA-A, B, C, and DRB1, and then considered the following donor factors for matching: age, sex parity, CMV status, and then other HLA loci, DQB1 and DPB1 TCE permissive mismatching, again using the original algorithm. As far as results go, donor parity, CMV status, HLA-DQB1 matching, and HLA-DPB1 TCE permissive matching were not associated with a greater than 1% improvement in overall survival or event-free survival. And this was the threshold that we used for clinical relevance. So if there was not an increase in mortality at 1 year or at 3 years post-transplant, or 1% increase in mortality, we set those factors aside. Again, donor age was important for overall survival and event-free survival. Donor sex was important for event-free survival, and I'll touch on that. But these had clinical impacts on outcomes and were considered for donor optimization. So when we consider the donor age impacts, donor age was more important for overall survival than event-free survival, and generally younger donors 18 to 30 are optimal, and I will show that data. As far as donor sex goes, donor sex had very little, if any, impact on overall survival, suggesting that any donor sex does not impact that. There was an association with event-free survival, but that was really tied more to likely size disparities that increased the risk of graft failure in the population. So looking at the impact of donor age, and again, that zone of, we called it the zone of indifference in this paper, or we can call it the zone of clinical relevance as well. But when we're looking at thresholds of donor age, once you get to about a donor age of 31, we start to cross over this threshold of the zone of indifference, suggesting that above the age of 31, donors are about the age of 31 and above, that donor age has an impact of about a 1% decrement in overall survival. That increases to about 2% once you get up into the 40 range, and this is all in comparison to a donor at age of 18. So suggesting that donors 19 to 30 are the optimal choice, or 18 to 30 are the optimal choice in that setting. Now, there have been some, I mentioned the paradigm shift in GVHD prophylaxis strategies. Now, PT-Cy, or post-transplant cyclophosphamide, given on days 3 and 4 post-transplant, has been extremely common for well over a decade in the haploidentical setting. And we did a study through the CIBMTR Immunobiology Working Committee that was led by Efren Fuchs that demonstrated that HLA-DPB1 TCE non-permissive mismatches influences overall survival and disease-free survival. This was a very interesting finding from this analysis, and so we wanted to carry that forward and better understand that in the unrelated donor setting, now that we are starting to see much more use of post-transplant cyclophosphamide. So a similar study team that was led by Shannon McCurdy and Efren Fuchs conducted this study through the CIBMTR Immunobiology Working Committee, focusing on the impacts of PT-Cy, as well as in the and the calcineurin inhibitor setting, and focusing on GVHD-free, relapse-free survival. So this does incorporate acute GVHD, but acute GVHD grades 3 to 4 is a fail. So it is survival in the absence of relapse, in the absence of acute GVHD grades 3 to 4, and in the absence of relapse and moderate to severe chronic GVHD. These were U.S. patients receiving transplant from 2015 to 2020 for our primary indications for transplant in the U.S., ALL, AML, and MDS. HLA-DPB1 TCE mismatching using the 2.1 algorithm was not associated with any outcomes in the PT-Cy setting. And that's demonstrated here for Griffiths, where we do not see an impact in PT-Cy. But I'd also argue that we did not see a difference in Griffiths in the CNI setting, represented here by methotrexate and tacrolimus as GVHD prophylaxis. So in the CNI setting, non-permissive mismatch associated with increased risk of acute GVHD 2 to 4 only. So not even severe acute GVHD. There was also a recent study in the EBMT that was led by Dr. Arrieta-Bolanos, Esteban, that also did not see an effect of HLA-DPB1 TCE mismatching in the PT-Cy setting. Now, we have updated our donor selection guidelines through the NMDP and CIBMTR, and I just want to pull out one of those tables from this JTCT publication earlier in the middle of last year. And so in the CNI setting, we are still suggesting that you avoid DPB1 non-permissive mismatches in MUDs, if possible. But in the context of other donor factors, such as donor age, really prioritizing younger donor age ahead of HLA-DPB1 non-permissive mismatches. In the PT-Cy setting, because we should be considering all donor types, we incorporated some detail on donor selection in the haplo setting, but in the unrelated donor setting, that we really just do not have evidence that DPB1 TCE non-permissive mismatching is associated with better outcomes. So being able to set that aside and maybe not consider that as stringently. So let's turn quickly to these two cases. So in the CNI setting, we have a 59-year-old non-Hispanic white female with high-risk AML. In CR1, the patient has underlying cardiac comorbidities that contraindicate use of cyclophosphamide for GVHD prophylaxis. They happen to be adopted, have no known biological relatives, and an unrelated donor search identifies several options. So here are our options, and I will go through these and give you my recommendations. So first, you have a 45-year-old male that's 8 of 8, DQB1 matched and DPB1 TCE permissive. However, they have a donor-specific antibody at a 5,000 MFI to a mismatched DPB1 allele. I would take that donor off the list immediately. You have other options that you can select. So the next options are a 25-year-old 8 of 8 female MUD, DQB1 match, and DPB1 TCE non-permissive mismatch. They also have no relevant DSA. Your next option is a 35-year-old male DQB1 mismatch, DPB1 TCE permissive, and no relevant DSA. In this setting, based on the findings that we've seen on the importance of donor age in the calcineurin inhibitor setting, I would prioritize the 25-year-old female donor for that purpose. If they're not available, the 35-year-old A to 8 male is a great secondary option. Now looking at the PT size setting, so here we have a 65-year-old non-Hispanic white male with high-risk AML and CR2. Allo-HCT using a PT-side GVHD prophylaxis strategy is planned, and they have no siblings or biological children. Keeping this pretty simple, so we're focused on the unrelated choices. An unrelated donor search identifies several available options. Which would you choose and why? And I'll go through this again. So again, we have that donor-specific antibody. I would take it off the list, especially if there are other options, even with a donor being quite young, 23 years old. It's just the additional treatment that's required to desensitize a patient delays transplant and can lead to additional complications. Just move on to your other options. Next option is a 30-year-old 8 of 8 female, DQB1 matched, DPB1 TCE non-permissive mismatch, and no relevant DSA. And then the next option is a 40-year-old male, 8 of 8, DQB1 match, TCE permissive, and no relevant DSA. Again, I would select the younger female donor. In this setting, if they're not available, the 40-year-old male may be a good option as a backup. And so with that, just to close, HLA-DPB1 TCE impact in the CNI-based GVHD prophylaxis setting, impact on outcomes appears to be limited to acute GVHD in the contemporary CIBMTR analyses. Esteban did show data related to relapse. I feel like that does need some validation in other data sets and is definitely worth pursuing. Relapse is a concern in malignant transplant for malignant disease. Also, prioritizing TCE matched or mismatched when all other donor factors, especially younger age, are equivalent. In the PT size setting, there is no evidence at this point, either in the EBMT datasets or in the CIBMTR datasets, to suggest there is an impact of TCE matching or permissiveness in the PT size setting. And findings in the haploidentical donor setting did not appear to translate into the unrelated. So with that, I'll go ahead and close and just want to acknowledge all the transplant centers who support the mission of the CIBMTR, providing data, and patients who enable us to learn from their their journeys, as well as our funding sources. So I will go ahead and stop there and turn it over to Dominic. Thanks.
Thanks, Steve. Yes. Then to you, Dom.
Okay. Can you see my slides okay?
Yes, perfect.
Yes.
We do have a little screen at the bottom, though, with our pictures. Maybe you'd like to move that out of the way.
Yeah, okay.
That's fine.
Okay, I'd like to thank the WMDA for inviting me to give this demonstration on the IPD HLA DPB1 T-cell epitope tool. First off, I'm going to start with a bit of background and history on the tool, which is an implementation of the DPB1 T-cell epitope algorithm developed by the University Hospital Essen. And this tool has been developed in collaboration between IPD and Essen. The first version of the DPB TCE tool was released in 2012, and this was based on cross-reactivity patterns of alloreactive T cells, but this limited the assignment of TCE groups to the proteins included in the original study. Later, in 2015, version 2 of the tool was released, and this was able to make in silico predictions of TCE groups based on functional distance scores, and that allowed TCE assignment to all alleles in the database. Most recently, in 2025, versions 2.1 and version 3 were released in April. Version 2.1 uses the same TCE group assignment as version 2, but now includes bidirectional immunogenicity. This is also included in version 3, which additionally splits the largest TCE group 3 into core and non-core alleles. This graph shows the usage of the different TCE tools over the last 18 months. And at the start of 2025, due to low numbers of users and with anticipation of the release of new versions of the tool, we retired version 1. Since then, we released in April version 2.1 and version 3, and at this time saw an increased interest in the use of these tools. Version 2 is still currently the most widely used. However, version 3 has seen the biggest increase since then. So I'm now going to give a demonstration on the use of version 3 of this tool, but what I'm going to talk about applies to the other versions as well. But before I do, there are just a few caveats I want to go over. So to use the tool, the HLA-DPB1 typing for a patient and a donor is needed, and this typing must be to 2-field resolution of the nomenclature or greater. Alternatively, you can use P groups or G groups. All the DPB1 typing you're using should be to the same resolution level. And as Esteban said, the predicted permissiveness may differ between different versions of the tool, particularly between version 2 and versions 2.1 and 3. And this is due to the inclusion of bidirectional immunogenicity. With that out of the way, using the tool is hopefully quite straightforward. Simply enter your patient and donor typing. Here I'm using 2-field resolution. And if you want to query multiple donors for a single patient, you can click Add Further Donors and then enter the additional information. For this demonstration, I'm going to stick with just one donor, and you can click Submit Query. And then the results of your query are shown below the input form. The results return the different allele typing that was provided by the user, along with their associated TCE groups and a prediction of the permissiveness. In this case, this is non-permissive in the host versus graft. The way the tool works is that it generates a unique URL for each query, and this contains the information used for the query. This URL can then be saved or sent to a collaborator to regenerate the results of the query. I've got another example here, this time with a null DPB1 allele, and in the case where a single null allele in an individual, the tool assumes that the individual is homozygous for the expressed allele. So now I'm going to talk about the application program interface, or API, that powers the tool. And an API is a piece of software that allows different programs to communicate with each other using a defined syntax. So in the examples I just gave, the web tool communicates the user's query to the matching API, which actually carries out the prediction and returns the results to the web tool. However, users can interact with the API directly and programmatically, and this interaction is made by an HTTP GET request. The results of the queries are then returned in JSON format, and for more documentation on using the API can be found on our website. So this is an example of a response to the API. And here, again, you have the patient and donor DPB1 type information, as well as their associated TCE groups, and a result, which is the TCE prediction. And the use of APIs means that people can query the database from within their own systems and in their own way. And I'm now going to run through a few ways that the API can be used directly. One way is on the command line using a basic command like curl or wget. However, this may make it difficult to read the JSON formatted response. An alternative way, and my preference, would be using a programming language like Python. This is a simple Python function which can get to the permissiveness, generate a prediction of the permissiveness given patient and donor typing. And in this way, you can access just the information from the response, the TCE prediction, without everything else. Using this simple function and some additional Python codes, just as an example of how it can work, I wrote a small script which was able to pass an Excel spreadsheet and generate another spreadsheet, including the TCE assignment for each of the pairs, as well as their matching status. And this piece of code was able to process 10,000 pairs in less than 25 minutes. The real power of APIs, though, is their flexibility and their ability to be used in different systems. And you can even query our API directly from an Excel spreadsheet. This can be done using the web service Excel function. Unfortunately, this function is only available, though, for Windows. In this example, the top right cell contains the formula for querying the API and extracting the result. And you can drag and drop the formula the same as you would with any other spreadsheet. And Excel can then take the typing information for the row, query the API, and give you a response. I will say it won't be as instantaneous as this. It may take a second or two for each row. And if you're analyzing hundreds or thousands of pairs, I would potentially recommend interacting with the API using a programming language like Python. So those are some of the ways that you can interact with the DPB TCE tool through the web interface with point and click, and directly through the API in multiple systems. For any information you'd like, more information you'd like on the use of this tool, please see the documentation on our website, or you can contact us by email. And I would like to thank my collaborators on this project, particularly James and Esteban, for their contributions to this work. And we are also preparing a manuscript on this tool, which goes into more detail about how the tool works and how we validated its output. And hopefully this will be available soon. And then lastly, I would like to thank you all for your attention.
Great. Thank you so much, Dom. And of course, to Esteban and Steve as well for your brilliant presentations. Unfortunately, we've run out of time for questions. I see that there's a couple of hands going up right now. Maybe if our speakers have a few more minutes, do you want to stay on for one or two questions?
Sure.
Sure.
Okay.
I'm happy to stay.
So the first question was for you, Esteban. You mentioned other permissive mismatch apart from core, non-core. Can you maybe elaborate on what you meant with that?
Yeah, so this is essentially, so the core non-core classification applies to all patient-donor allele combinations where you have a mismatched group 3 allele. If that is not the case, then that's what we would consider the other permissive. So in those cases, we don't have any subclassification of mismatches. It's just a permissive mismatch that involves probably an allele from group 1 or group 2. Right, so it's the other group. It's the, as you saw, it's the least frequent subgroup. And so it makes it difficult to delve deeper into what differences might be in there. But I don't expect the differences to be as big as for group 3 because of the diversity that you saw before. That was really a feature of group 3 that allowed us to perform this subclassification.
Thank you. A follow-up question then is that if you want to reduce the risk of relapse, would you rather look at non-permissive or non-core permissive mismatch?
Well, that's a tricky question. As Steve mentioned before, this probably requires validation. And also, we have to point to the fact that those findings were not obtained under PTSI. So we would have to see what happens there. But going back to what we observed, I can tell you that the non-permissive mismatches consistently increased non-relapse mortality. And this in the EBMT cohort was not the case for non-core permissive mismatches. So to me, that points to the fact that non-core permissive mismatches might be that sweet spot between immunogenicity and the detrimental effects of GVHD. But with all the caveats said before, this is something that has to be taken with a pinch of salt. What I can say is that the behavior was not the same between non-permissive and non-core permissive mismatches.
Thanks, Esteban. Looks like there's still quite a bit of work for you and your team to do then. Then maybe this question is for you, Steve. It's a long question, but I think it really boils down to non-malignant disorders. Do you prioritize core permissive mismatches in the context of non-malignant disorders?
That's a great question. Thanks, Alberto. And something where we need, frankly, more numbers to better understand that. In the studies, though, where we were looking at developing a donor selection score, we have included malignant and non-malignant diseases, at least in the original study that we did that was published in 2018. And in that case, donor age was more important. So I would be prioritizing younger donors in that setting, but then also thinking about the way that we've shifted our paradigm for GVHD prophylaxis. That most of that experience has been described in the malignant setting. However, there is great emerging data in sickle cell disease and severiplastic anemia demonstrating very effective acute GVHD and chronic GVHD mitigation in the setting of haploidentical transplants with PTSI. My expectation is that that will continue, and we will see those similar effects in mismatched unrelated donor transplantation as well as fully matched transplantation in those settings. So stay tuned for that, but I think the changes that we've seen in the way that we're actually performing these transplants will likely have a larger impact at the end of the day. And to date, we really haven't seen an effect in the PTSI setting of DP mismatches. So more to come on that, but definitely a great question and something we need to be considering going forward.
Brilliant. Thank you so much. That is the end of our Q&A. Thank you for everyone that hung on for the Q&A and also for all of the presentations. Thanks for your time today, even though we ran over a little bit. As I mentioned, the session was recorded and you will be able to watch it back. We will post it on Share sometime in the next 24 hours, along with the slide deck. And then finally, a big thank you once again to our presenters, Steve, Dom, and Esteban. It was brilliant to tap into your knowledge. Also, thank you so much to my colleague Hans-Peter for assisting me in the moderation. Please keep an eye out for future educational webinars. And lastly, a big thank you to our sponsor of this webinar, NextGen. Until next time, everyone, thank you and see you again. Bye.
Thank you.